Hello, this is my capstone project for Fine 204 during the spring 2020 term. I thought it would be an interesting idea to paint a polychrome real-world setting through the eyes of colorblind individuals. I will paint the scene in normal color vision as well as 3 different types of color deficiencies. I would also like to mention that I will be discussing human color vision and dichromacy. As I'm showing my work, I'll be discussing the anatomy and genetics behind color blindness since I'm a biomed student and I find it super fascinating. So here are the timestamps if you want to jump ahead or to go back to anything. I uploaded this video to YouTube so it's easy for you to speed it up if it's too long. Okay, let's get started. I drink a lot of kombucha, so I've been saving glass bottles for this assignment. I have 4 bottles to represent 3 colorblind views and 1 normal view. I've cleaned the insides and I'm putting a layer of white on top. Color blindness, also known as color deficiency, is a group of conditions that affect the perception of color. They have trouble distinguishing between shades of color. Color is perceived at the back of the eye, or the retina, using 3 cones that are sensitive to a specific range of color. Normal trichromatic vision uses all 3 cones. Color deficiency is a malfunction or the absence of 1 or more cones. I chose my carpet as a polychrome setting because it has a nice range of colors and didn't change too much throughout the day. There are 3 types of cones at the retina that differ in photopigment or cone opsins. Each photopigment is sensitive to a different range of wavelengths of light. The S, M, and L cones are associated with the short, medium, and long wavelengths respectively. When the appropriate wavelength hits the photopigment, it causes the cone to fire an actual potential, which then sends a signal to the brain. Each type of cone can fire at a specific range of wavelengths, but are more likely to fire at their peak absorbance. A collection of firing cones at a specific rate allows the color to be perceived. Let's get to mixing colors. I made my palette into a grid to represent the 7 main colors in the carpet and the 4 different types of vision. The top row is of normal color vision, while the other 3 rows will represent different types of color deficiency. The 3 other bottles will represent dichromacy, or the use of only 2 cones instead of 3. The other rows require me to filter out the colors not seen in dichromats. However, this does not restrict which paints I use. This just means I have to use less of it. I'm basing my estimations on the absorption spectra as seen in the diagram. The second row represents protonopes, or those who cannot use their long L cones. They rely on S and M cones to see color, so reds would not be visible, and would instead appear gray. Purples cannot be distinguished from blues, and oranges would shift to a dim yellow. The third row represents deuteranopes, or those who cannot use their medium M cones. They rely on L and S cones to see color. Their vision is similar to protonopes, but without dimming the hues. This is because the absorbance curve of M slightly overlaps with L cones. The fourth row represents tritonopes, or those who cannot use their short S cones. They rely on M and L cones to see color, so blues would appear greenish, yellows and oranges would appear pinkish, while purples are a deep red. As I am color blocking the bottles, let's go further into depth about the eye and its connections to the brain. A visual field is picked up by visual receptors called rods and cones. They are long bipolar neurons that directly send a signal to the brain. As previously mentioned, there are cones at the retina that are used for color vision. They are abundant in and near the fovea and are most active in bright light. Rods are a different type of visual receptor that are abundant in the periphery of the human retina. They respond to faint light and are used to clarify an image. Both rods and cones contain photopigments that release energy when struck by light. Cones consist of a chromophore called 11-cis retinol. It is bound to a transmembrane protein called opsins, which modify the pigment's sensitivity to different wavelengths of light. A specific wave of light causes 11-cis retinol to change to all trans-retinol, thus releasing energy that activates second messages within the cell and causes the transmission of a neural signal to the brain. Here I am adjusting the color and the patterns to the carpet. as well as compensating for the shadows and lighting. Protonopes and deuteranopes are collectively termed as red-green color vision defects, while tritonopes are termed yellow-blue color vision defects. Protonopes have malfunctioned L pigments, and deuteranopes have malfunctioned M pigments. Protonopes and deuteranopes are coupled together because their opsins share 98% amino acid sequence identity, compared to 43% identical to S opsin. In most cases, the cause of red-green color blindness is the loss of the genes that encode the photopigment. Photopigments M and L genes are on the X chromosome. This means that it is a sex-linked trait. It occurs via the X-linked recessive inheritance. Men carry X and Y chromosomes, while women carry two X chromosomes. If there is mutation on the X chromosome in men, they will be colorblind. Women will be colorblind only if both X chromosomes have the mutation. If only one has a mutation, she is a carrier. It is more likely for men to be colorblind because they only have one X chromosome. Tritonopia is a blue-yellow color blindness caused by the malfunction or loss of the genes that code for the S opsin. Unlike M and L opsin, the gene coding for the blue receptor lies on chromosome 7. Therefore, it is not sex-linked, and it is found equally in both men and women. It is a dominant characteristic with incomplete penetrance. 3 amino acid substitutions in the transmembrane domain of the S opsin are known causes. These are the main reasons for these types of color blindness. There are many causative mutations that can cause color deficiency. It can originate from at least 19 different chromosomes on 56 different genes. The ability to perceive color is not solely based on the photopigment, but the ability of the cone to function and relay the message to the brain. In conclusion, here is what the final product looks like. The bottle to the far left represents normal color vision. The second bottle represents protonopia. The third represents deuteranopia, while the far right represents tritonopia. The end.